Okay, I think we'll get going because I don't want to keep you unnecessarily. Right, so we are recording now. Okay, so yesterday we looked at the light-dependent reactions, and today we're going to look at the light-independent reaction, which is also known as the Kelvin cycle. And I'll tell you a little bit about Kelvin himself. So just to start with, let's just do a little bit of a recap from yesterday. So can somebody shout out the two parts of the chloroplast?
Stroma,
good.
Membrane.
Yeah, good. So the stroma and the thylakoid. Okay. Next one. So let's see. Can you think of everything? I think there's 5 things that go into it. What goes into the light-dependent reactions? Go on. Light. Somebody just shout out light. Yeah. What else?
Water.
Water. Good. What's the thing that carries the hydrogen and the electrons?
ADP.
Well, we need ADP. We need as well, but that's not
chlorophyll.
NADPA.
No.
Yeah,
NADP.
Yeah, and I think that was it actually. Yeah, 4. Yeah. So water, light energy, ADP and inorganic phosphate to be built up into ATP, and oxidized NADP. What are the outputs then? What comes out?
Oxygen.
Oxygen, yeah.
Reduced NADP.
Reduced NADP, and the energy in... How does the energy...
ATP.
ATP, right. So oxygen, ATP, reduced NADP. So, I mean, I generally do, for photosynthesis and respiration, I generally break it down into stages and then try to get you to remember what goes in and what comes out of each stage. And what comes out largely goes into the next stage. So oxygen's thrown away, but the ATP and the reduced NADP are needed for the Kelvin cycle, along with carbon dioxide. What's the word that means splitting water?
Photolysis.
Well done. Photolysis, yeah, photolysis, but photolysis is the correct pronunciation, I think. And why is water necessary? What does it supply?
Hydrogen.
Yeah, it supplies hydrogen to reduce... And electrons for what? There. So hydrogen to reduce NADP, electrons to replace the electrons which are lost by PS2, because they go to PS1, and PS1 electrons are picked up by NADP. And what's the name of the chain of carrier molecules that take the energy from the electrons and build up ATP? Begins with electron.
Electron transport chain.
Brilliant. Right. Okay. I mean, those, if you can remember the answers to those questions, I think that's pretty much most of what you need to know about yesterday. So let's move on. And I want to play this again. And I know you've seen most of it. So the last 2 minutes talk about the Kelvin cycle. So I think it is worth just watching it both as a recap and just to take it on to the final stage. We haven't got the commentary on this bottom. So right, through the stoma. These are the spongy mesophyll cells travelling through an airspace in the leaf towards the palisade mesophyll at the top of the leaf, which is where most photosynthesis occurs. Into the cell. There's endoplasmic reticulum. There's the nucleus. There's the purple things are the mitochondria. The green things are the chloroplasts. Okay, so there's a granum, stack of thylakoid membranes, and the space in between is the stroma. So the surface of the thylakoid membrane is where the electron transport chain takes place, and those molecules embedded in the membrane there are the carriers for the electron transport chain, and the other bits which are spinning around are the enzyme ATPase, which is where ATP is built up. So the energy on the electron transport chain the energy from the electron transport chain is transferred to the ATPase effectively to build up ATP. And we'll look at this in a bit more detail when we do respiration. Actually, I'm getting a bit confused because we did see the whole of this yesterday. And those little wigglies are the ATP molecules that have been built up. And as it said in the commentary, this has all been slowed down about 1 million times, so it would be happening a lot more quickly now. Okay, so this is the objective for today. Understand the light-independent reactions as reduction of carbon dioxide using the products of the light-dependent reactions, carbon fixation and Calvin cycle, the role of GP, GALP, RUBP, and Rubisco. Most of what you need to remember here is about just these names. It's the terminology. It's not what happens is fairly straightforward. Carbon fixation just means taking carbon dioxide and turning it into carbohydrate. We'll come on to the GP, GALP, RUBP, and Rubisco in a little while. And secondly, know that the products are simple sugars that are used by plants and other organisms in respiration and the synthesis of new biological molecules. So glucose really and the photosynthesis is the beginning of everything that plants do. Plants have to make all their own molecules. If consumers like us, if we need a particular molecule, a protein, we go and eat it. But plants can't do that. They have to build every fat, every amino acid, every polysaccharide from scratch. Okay, I want to start off with Melvin Calvin, which I thought was a great name. I've known about the Calvin cycle for lots of years, but only last year did I find out that his first name was Melvin, and I just found it amusing. So I thought I'd just give you a little bit of a bio for him. I think he looks a bit like Mr. Hardison, actually. Anybody think that?
Who's that?
Anyway,
Head teacher.
I don't know.
You don't know Mr. Hardison? He's the head teacher, yeah.
Oh, him.
Anyway, so in the 1950s, so it's quite a long time ago, all this is worked out, and he was basically in charge, but he worked with these other two guys called Bassam and Benson to look at the reactions that took place in the stroma of chloroplasts. And this is now known as the Calvin cycle or the light-independent reactions of photosynthesis. Although there are some scientists who think that he was a bit mean because he kind of denied any credit to the other two people who actually did quite a lot of work in it. What he did, you don't need to know this little bit, but just out of interest, he used this unicellular alga called chlorella, and he gave it carbon dioxide with radioactive carbon-14, and then he gave it to them, and then shortly after that, I think he dropped them in liquid nitrogen or something to stop any reactions, and then he analysed it to find out what products had been made, and he did this in kind of shorter and shorter time periods after supplying the radioactive carbon-14, and he was then able to identify the chemical substances that were produced. Yeah, Calvin he's not meant to be a very nice chap, actually. So James Benson worked with him for 7 years, but then Calvin sacked him, and when he wrote his autobiography, he never even mentioned his name once, which is slightly unpleasant. And if you in 61, Calvin was awarded the Nobel Prize, and the other two didn't get anything. A a bit like about the story of DNA, isn't it? With what's her name? Who's the woman who never got any credit?
I can't remember her name.
No, I can't. Rosalind Franklin.
Yeah, give me the name.
Yeah, if you're interested, anyway, there's a link there to a program about him, but don't it's not on the spec, that, so you don't need to worry. Right. These are basically the terms you need to know for this. Obviously, CO2, ATP. We've done that before. We've also talked about NADP becoming reduced NADP, and you will see it written sometimes as NADPH. That is exactly, it's just an alternative way of writing it. Reduced NADP is the same thing as NADPH or NADPH2, because it actually, I think, holds 2 hydrogens. If something is American, they tend to use the NADPH, but I think the exam board use reduced NADP, so I would prefer you to use that, although it's not wrong to use the other one. Right, there was another product called ribulose bisphosphate, but the good news is you can write RUBP. RUBP is a molecule with 5 carbons, which carbon dioxide attaches to. So that's the carbon fixation, is carbon dioxide joining to RUBP. And then we have another extremely long one, ribulose bisphosphate carboxylase. What sort of product is that? Ending in A's?
An enzyme.
It is an enzyme, and that is the enzyme that catalyzes the addition of carbon dioxide to RUBP. So ribulose bisphosphate carboxylase, it's adding CO2 to RUBP. But again, that you can just use Rubisco. That's absolutely fine. And it is said, probably true, that Rubisco is the most common enzyme on the planet, because every organism that photosynthesizes has it. And then there are two products which also feature in the Calvin cycle, which is GP and GALP. And GP is glycerate 3-phosphate. GALP is glyceraldehyde 3-phosphate. These are both carbohydrates, and GALP, I think, is a sugar. Okay, so GALP is then turned into glucose, and finally at the bottom, carbon fixation or carboxylation is just the adding of carbon dioxide, as we've said. Okay, so we'll have a look at the Calvin cycle. So first of all, CO2 combines with a 5-carbon compound, RUBP, but that makes a 6-carbon compound, but nobody has ever been able to isolate that 6-carbon compound. So Calvin and Benson and Basson weren't able to find this. So that's led people to conclude that basically it's unstable, and as soon as it forms, it breaks down into 2 3-carbon compounds. And the 2 3-carbon compounds are converted to a 3-carbon triose phosphate by reduction. So that's adding the hydrogen from NADP. So that's where the NADP comes in, and being given energy from ATP.
Sir?
Yeah.
Why did they want a 6-carbon compound?
Well, because if you're starting with a 5-carbon and you add CO2, you have to really form a 6-carbon compound before you can get 3-carbon compounds. The other alternative is to say the 5-carbon compound splits up into a 3 and a 2.
But why were they trying to do it? What were they trying to achieve?
Well, just to understand the stages in the cycle.
Oh, okay.
Okay. Okay, so the triose phosphates then make glucose and are also recycled to make more of the RUBP. Okay, and that's it. So if we look at the, it's a bit easier to see graphically, I think. again, this is taken from the book, and I've kind of broken it down into 3 stages. So the first stage here is the carbon fixation stage. So the 5-carbon compound is ribulose bisphosphate, RUBP. Carbon dioxide is added to that. That gives us 6 carbons. Now that forms 2 3-carbon compounds, which is GP. And that's the stage that involves Rubisco, the enzyme. The next bit is reduction, and that's the bit where the products from the light dependent reaction come in. That's where reduced NADP and ATP are needed to add phosphate and to add energy, and that produces GALP. And then the third stage is regeneration of, because you want to make more of the 5-carbon compound RUBP in order for the cycle to continue. So most of the GALP actually goes back round the cycle to make RUBP, and more ATP is needed for that stage as well. So every 6 carbon atoms, sorry, once you've had 6 carbon dioxides added to the cycle, so in other words, you've kind of been around 6 times, then you can form a molecule of glucose. Okay, but 5/6 of the carbons get recycled every time. What would happen if the light dependent reaction stops? Where will the cycle stop? Do we understand the question?
Stage 2.
Well, you won't have, if the light dependent reaction stops, you won't have reduced NADP and you won't have ATP. So whoops, just lost my pointer. I'm just going to change my pointer. So you won't have these 2 reagents here, so basically this stage won't happen, so you'll get stuck at this thing. It should, carbon dioxide will still be able to combine to produce the 3-carbon PG, but it won't continue any further than that. Right, this is a diagram from the book. This gives it in a lot more detail. So let's just take that round one more time, because what they've done here, they've added, as you can see, 6 carbon dioxides. That's to show you effectively the making of 1 glucose. So carbon dioxide combines with RUBP, catalyzed by Rubisco. Secondly, you form a 6-carbon compound. 5 + 1 is 6. That breaks down to 2 3-carbons. So if we've got 6 carbon dioxides, we'd actually have 12 lots of GP then. Okay, so that's 36 molecules we've added. Then we've got reduced NADP and ATP from the light dependent reaction, and that turns GP into GALP. Some of those GALPs or 2 of the 12 GALPs get turned into glucose, hexose sugar, 6 carbons. The other 10 get recycled with the help of energy back into more RUBP, so that more carbon dioxide can be fixed. Is that okay? Anybody got any questions about that?
Where does the RUBP come from?
Well, it's always present. So when the chloroplasts are made, I mean, it's one of the proteins, or it's one of these substances which will be coded for in the genes of the chloroplast.
So it's just a protein that can be combined with.
Yeah, it's not ribulose bisphosphate. It's a phosphorylated protein. So a protein that's had 2 phosphates added to it. But when the chloroplast is formed, the genes on the chloroplast and on the nucleus of the cell will make RUBP. So it will be there, and as it's used up in the cycle, it's regenerated, so it never is depleted completely.
And wait, what is the RU-BISCO? Where's that used again?
Rubisco. That's effectively here. So it's joining. That's what catalyzes the fixation of carbon.
Okay.
These two together. So it's this stage which is catalyzed by Rubisco.
So it sort of joins them together.
Yeah.
Okay.
I mean, it's what sort of reaction? Joining reaction in biology?
Condensation?
Absolutely right. Yes. So it's a condensation reaction. So water will be produced as well, and it's catalyzed by Rubisco. But it's just because this idea of fixing carbon dioxide is really important. That's why Rubisco kind of gets a big press. I mean, there will be enzymes involved in this, all the other stages as well. But it's this fixation of carbon that's really important. Partly because it takes carbon dioxide out of the air, relating back to climate change and everything to do with that. But also because that's where all the food in the world comes from, effectively, is that carbon joining onto RUBP and making GP and GALP and then glucose. Okay. Right. Okay, a few questions then. See, test your memory. Can anybody name a substance that has been phosphorylated? Let's go back. What substance has been phosphorylated there?
I can't remember what phosphorylated means.
Well,
ADP.
Well, ADP hasn't, because it's going the other way. ADP.
Oh,
So phosphorylation means adding phosphate. Just think about the name. There is only one stage where... No, there's two, actually. There are two answers to this. If ATP is broken down, what's happening to the... If you look...
It's GP, isn't it? It's
GP and GALP.
And then GALP.
Well, right. If you look here, look at this. Here you've got 12 ATPs are turning into 12 ADPs and 12 inorganic phosphates. Here, 6 ATP are going to 6 ADP. Now, why is that different? It's different because what happens to the inorganic phosphates here? They get added into RUBP. These ones don't get added in. They're still separate. Do you understand the difference? So these are shown as inorganic phosphate because they're not added to GP. But there is no inorganic phosphate because that has been absorbed into RUBP. So really, the answer is GALP has been phosphorylated into RUBP. This is the answer in the book. Oh, it has been phosphorylated. Yes, it has. So the GALP is phosphorylated into RUBP. Right, substance formed by reduction. What's been reduced here?
Is it the GP?
Yeah,
oh, no.
GP is reduced. It's had hydrogen added to it because, again, the hydrogen has been lost from here. Reduced NADP becomes NADP. So hydrogen is given to GP to become GALP. So a substance formed by reduction will be GALP. And why does the diagram show 6 molecules? Well, we've talked about that. It's the minimum number to make 1 molecule of glucose. And each CO2 contributes 1 carbon and 6 are needed for each glucose. Right. Why is glucose so important? Number one, it's used for respiration in every plant cell, and pretty much most living cells use it. Secondly, it's made into sucrose and transported in the phloem, and you'd have done that in topic 4. What has to be added to glucose to turn it into sucrose?
Fructose?
Fructose, good. Oh, there you go. And it's also polymerized to make starch and cellulose. So alpha glucose, polymerize it, you get starch. Beta glucose, polymerize it, you get cellulose. And cellulose makes up something like 50% of the mass of a plant. So not a tree, but of a normal, a non-woody plant, has a lot of cellulose because of all the cell walls. Right, so starch is energy storage. Cellulose makes up the bulk of the cell walls. And it's the starting material for lipids, amino acids, therefore proteins and nucleic acids as well. So glucose is the starting point for pretty much every molecule that the plant makes. What else do you need? What has to be added to glucose to make a protein? What mineral, what element or mineral ion? Glucose is carbon, hydrogen, and oxygen. What else do you need to make an amino acid? Think of the amino acid structure. What does amine?
Oh, it's a type of group, isn't it?
Yeah, an amine or an amino group has what though? What element?
Nitrogen.
Correct. Well done. Is that Jeff? So nitrogen. Amine group is NH2, and plants need nitrates in order to get nitrogen to build proteins. What else is in nucleic acids? Think of the structure. What's the backbone of a nucleic acid? It's
Phosphate.
the sugar and
Phosphate.
phosphate. So what element is in phosphate?
Phosphorus.
Phosphorus. So plants need phosphorus again. So they all need nitrogen. Some amino acids need things like sulfur. They all need nitrogen for the nitrogenous bases, the 4 bases, and phosphorus for the phosphate groups as well. So how do plants take in those? Active transport. Hopefully that we're trying to bring together some of the different things you've covered in the last year. Right. Okay. This is the last slide. This is the one that we saw the first part of yesterday, and we'll watch this through, and then if you've got any questions, but apart from that, we're done.
In order for plants to grow, they need inputs of carbon dioxide, water, and energy. The chemical process by which plants use these resources to manufacture glucose, the building blocks of plants, is called photosynthesis. In the process, oxygen gas is produced as a byproduct. The energy for photosynthesis originates in the sun and arrives at the Earth as sunlight. This light has both a wave and a particle nature. The particles, or photons, are the smallest units of light. Photons oscillate along a path, which is measured as wavelengths. The light emitted from the sun contains photons in a wide spectrum of wavelengths, called the electromagnetic spectrum. Photosynthetic organisms use only a small portion of the electromagnetic spectrum, called visible light. Photosynthetic organisms contain pigments that facilitate the capture of wavelengths of light in the visible light range. The color of the pigment comes from the wavelengths of light reflected. Plants appear green because they reflect yellow and green wavelengths of light. Red and blue wavelengths of light are absorbed by these pigments and provide the energy that is used for photosynthesis. Within eukaryotic photosynthetic organisms, also known as photoautotrophs, the chemical reactions of photosynthesis occur within plant cells in specialized structures known as chloroplasts. Photosynthesis consists of 2 sets of reactions, the light-dependent reactions and the Calvin cycle. Within the chloroplast are small disc-like structures called thylakoids, which are surrounded by a fluid-filled space called the stroma. The reactions that synthesize glucose, the Calvin cycle, occur in the stroma. The light-dependent reactions occur in the thylakoid. It is here that conversion of light energy to chemical energy is initiated. In most photosynthetic organisms, thylakoids contain pairs of photosystems, called photosystem 1 and photosystem 2, that work in tandem to produce the energy that will later be used in the stroma to manufacture sugars. The photosystems of the thylakoid consist of a network of accessory pigment molecules and chlorophyll, the molecules that absorb the photons of light. Within the pigment molecules, the absorbed light energy excites electrons to a higher state. Photosystems will channel the excitation energy gathered by the pigment molecules to a reaction center chlorophyll molecule, which will then pass the electrons to a series of proteins located on the thylakoid membrane. Photons of light strike photosystems 1 and 2 simultaneously. We will examine what happens with the photons striking photosystem 2 first. The energized electrons are passed from the reaction center of photosystem 2 to an electron transport chain. The electrons lost by photosystem 2 are replaced by a process called photolysis, which involves the oxidation of a water molecule producing free electrons and oxygen gas. While this oxygen gas is a byproduct of photosynthesis, it is an important input to the cellular respiration pathways. As electrons pass through the electron transport chain, the energy from the electron is used to pump hydrogen ions from the stroma to the thylakoid, creating a concentration gradient. This gradient powers a protein called ATP synthase, which phosphorylates ADP to form ATP. The low-energy electrons leaving photosystem 2 are shuttled to photosystem 1. Within photosystem 1, low-energy electrons are re-energized and are passed through an electron transport chain where they are used to reduce the electron carrier NADP+ to NADPH. When the chloroplast is receiving a steady supply of photons, NADPH and ATP molecules are rapidly being provided to the metabolic pathways in the stroma. Therefore, the ATP and NADPH formed during the light-dependent reactions are used in the stroma to fuel the Calvin cycle reactions. The Calvin cycle consists of a series of reactions that reduce carbon dioxide to produce the carbohydrate glyceraldehyde 3-phosphate. The cycle consists of 3 steps, the first of which is carbon fixation. In this step, carbon dioxide is attached to ribulose 1,5-bisphosphate, resulting in a 6-carbon molecule that splits into 2 3-carbon molecules. The second step is a sequence of reactions using electrons from NADPH and some of the ATP to reduce carbon dioxide. In the final step, ribulose 1,5-bisphosphate is regenerated. For every 3 turns of the cycle, 5 molecules of glyceraldehyde 3-phosphate are used to reform 3 molecules of ribulose 1,5-bisphosphate. The remaining glyceraldehyde 3-phosphate is then used to make glucose, fatty acids, or glycerol. It takes 2 molecules of glyceraldehyde 3-phosphate to make 1 molecule of glucose phosphate. Thus, the Calvin cycle has to run 6 times to produce 1 molecule of glucose. These molecules can remove their phosphate and add fructose to form sucrose, the molecule plants use to transport carbohydrates throughout their system. Glucose phosphate is also the starting molecule for the synthesis of starch and cellulose. Plants produce sugars to use as storage molecules and structural components for their own benefit. By utilizing the energy of the sun, along with inputs of water and carbon dioxide, plants act as glucose factories. Photosynthetic organisms are the primary producers of glucose on the planet. They also produce oxygen gas as a byproduct, and thus serve as the foundation of life, providing food and oxygen for the complex food webs on both land and in the oceans.
Okay. Right. So I'll stop the recording there.